Let us continue our discussion on fitness landscapes of antibiotic resistance. Welcome back, everyone. So, as we saw in the last time, in the last video, we have two variants of a gene in an E. coli. Both these genes are present in the nature. The differences between these two variants of the same gene are at 5 positions. In the made up sequences that we did, one variant had all A's and the other variant had all G's. And we saw that one variant had an MIC which was really low and the other variant had an MIC for a particular antibiotic which was 1,000-fold higher. And evolution from the low MIC to high MIC variant of the gene can only take place via acquisition of one mutation at a time. And these 1 mutations, because there are 5 options for acquisition of the first mutation, 4 for the second one, 3 for the third one, and so on and so forth, there are 120 different paths that are available for me to transition from the ancestral variant to the ecological variant, which offers 1,000-fold higher MIC. And these 120 paths can be classified into one of these two groups: fitness, where MIC is the proxy. In green is the path which is permissible in a Darwinian sense. So, green path is a one which is permissible in a what is referred to as a Darwinian sense. And what we mean by that natural selection will take this path. Natural selection will take this path. Because along this path, fitness or MIC is monotonically increasing. So, we say natural selection will take this path because along this path fitness, which is quantified in terms of MIC, is monotonically increasing. This class of paths is one particular group. Alternatively, we also have paths which belong to in this blue category. These are paths which are non-permissible in a Darwinian sense, non-permissible, and the reason is because fitness is not increasing. Fitness is not increasing along this path. As you can see, along this path, the first mutation increases fitness, the second mutation also increases fitness, but the third mutation actually leads to a decrease in fitness, as a result of which we have a local peak here. So, fitness is not monotonically increases. We reach a stage along this path where fitness increased from behind, but also going forward it is decreasing. So, through this local peak you move in either direction and fitness is decreasing, and hence you are stuck in that local peak because in a Darwinian sense you cannot decrease fitness, natural selection will not permit that. So, and then we asked that in these two groups, how are these 120 paths divided? And what we found out that a overwhelming majority of the paths were present in this particular group. There were only 18 paths which were present in this design, which was going to be permissible by natural selection. So, these 102 paths were the ones which had a local peak in them. Somewhere along their path there was a local peak, and hence these are not permissible. And all of this is a result of epistasis. And this is sort of the first manifestation that we see of epistasis coming into being. Remember that epistasis, if it wasn't there, then we would have, irrespective of our starting point, irrespective of the order in which the mutations were acquired, all paths would lead to the highest fitness. The highest fitness, in this case the ecological strain, into fitness being quantified as an MIC. However, because epistasis is playing out, it means that some mutations are beneficial in some contexts and some mutations are not beneficial in some contexts. Let us do this with an example. Let us imagine that we have a DNA sequence and we have two possible mutations on this DNA sequence. Hence, we have 4 possible genotypes. This is mutation A, also present here, and this is mutation B, also present here. Let us say that the fitnesses of these genotypes are 1, 1.04, 0.98, 1.09. If these are the fitnesses, then looking at these numbers and these mutations should tell us something. What we should do while analyzing this data is compare this ancestral sequence with this mutant. Let us call this ancestor, this is mut1, this is mut2, and this is mut12. Comparing the ancestor with mut2 tells me that the circle mutation, this mutation, when it happens in this background, because transitioning from here to here is essentially introducing this circle mutation in the black background. This mutation is a deleterious mutation. Because delta F associated with it is 0.98 minus 1, which is minus 0.02. Hence, this is a deleterious mutation. But if we look at the effect of same mutation, but in a different strain now let us compare mut1 and mut12. This is the same mutation occurring, but in a different background. So, this is the same circle mutation occurring, but occurring in the following background. When in not the plain background this time, but the plain background which is carrying this red mutation. In this case, the delta F associated with the blue background is 1.09 minus 1.04, which is 0.05. And this is a case where a mutation which was originally deleterious has actually become beneficial only because of the presence of this one other mutation. In this case, this mutation has now become a beneficial mutation. So, what we have here is epistasis dictating the nature of the mutation that is taking place in such a context. In one background, the mutation was deleterious, but presence of just one more mutation makes the same original mutation beneficial, transitioning from deleterious. And this is epistasis, and this is the reason why we have so many local peaks, because some mutations, their genomic context in which they are occurring is extremely important, and as a result of that, it is often that in a 5-step path, you will often get stuck in a local maxima and hence not be able to move forward. So, in this context, what has epistasis done? To begin the process, we had 120 mutations, not mutations, we had 120 paths. And if we did not know anything, if there was no epistasis, then perhaps each of these paths was equally likely. This is assuming if there was no epistasis. And there was a single global peak, and irrespective of starting point, you would converge to that global peak. However, we know there is epistasis, and figuring out this landscape by Hartle and co-workers told us that of the 120 paths, 108 actually can be ruled out. These could not have been the paths that nature took to go to the fitness peak, and hence the total number of paths that we have to worry about is actually, this is not 100, this is 102. The total number of paths that we have to worry about is actually 18. These are the only relevant paths. So, in some sense, epistasis reduced the number of solutions that was available to me from 120 to only 18. Viewed in such a context, it seems like epistasis has made evolution predictable or more predictable. Because if it was not acting, then I had 120 paths to worry about, all of them were going to be permissible, but because it is acting, then I do not have to worry about 120 paths, I only have to worry about these 18 paths which lead to increase in fitness. However, what we also must realize in this case is this was a special case where the end and the starting point were defined to me. This starting point of the evolution experiment, the ancestor lab strain and the ecological strain, they were defined to me. So, the start and end of this evolutionary trajectory were defined to me. However, when I am thinking of evolution, the start and end are not often defined to me. Very often the question I would ask is that I have this particular variant, and if I were to evolve this, what would happen? It is only because in this study access was available to this end point of evolution that I was able to go back and check, which would not often be available in the context of evolution being predictable or me trying to predict an evolutionary process. Nevertheless, this was an important study which showed how epistasis constrains adaptive movement, and of all the paths that are available for a population to move on, it only takes on a very few. It can only take on a very few. Let us now move to the next case of epistasis that we want to discuss, an experimental case of epistasis. This has a little bit of context before we start. So, I told you about Stephen Jay Gould asking this rhetorical question that what if we were to evolve the planet again, what sort of life processes would take place here and what sort of organisms would be inhabiting the planet if 4.5 billion years were allowed to happen again. And that is of course a rhetorical question to which there is really no answer. However, around the same time, a researcher in the United States started the following experiment, and the experiment is that E. coli is being evolved in an environment with low glucose. So, these E. coli species, these E. coli strains that are growing here in these flasks, they have low amounts of glucose as the only carbon source that is available to them. And this experiment was started in the late 1980s. So, what you are doing is you take E. coli in a flask and you give it low amounts of glucose and evolve in this condition for a long time. However, what the researchers did in this case back in the 80s was that they did not take one flask, but they took 12 flasks, all containing the same E. coli at the start of the experiment. So, you had an E. coli population in lab, these were all identical, and these E. coli were used to seed these populations, these 12 populations in 12 different flasks in identical conditions. That is another major advantage of doing evolution in lab, that you can just maintain the exact same environmental condition in something as diverse as these 12 different flasks. And in each of these flasks, they were evolved for a long time. In fact, we will spend a lot of time trying to understand major lessons from this experiment, because this experiment has become very famous now. It has a name of its own, and it is called the long-term evolution experiment. It is also referred to as LTEE. Let me write that again. It is referred to as LTEE. The experiment was started off in California, from there it moved to Michigan. Now, the person who started it has retired, so the experiment is now in Texas. From 1980s, it is still going on, and recently it has crossed 80,000 generations of E. coli. Remember that we discussed that from an evolutionary point of view, it is not the quantum of time. So, it is not important that 35 years or so have passed since the start of experiments, since the start of this experiment. What is relevant is that how many generations have been processed, and from the context of this particular experiment, we have processed about 80,000 generations in each of these 12 different independent flasks. These 12 different flasks are referred to as replicate lines. And in a video coming up soon, we will discuss how do we actually perform these evolution experiments that can go for tens of thousands of generations in a lab setting. So, we have 12 replicate lines. And obviously, this is a study that has been going on for such a long time, has given us remarkable insights into how evolution can take place, what are the molecular mechanisms which drive these evolutionary process, and so on and so forth. But from the context of today's video, what we want to discuss is just something that was observed in one such line. This is a paper that was published in Science and led by somebody called Tim Cooper. And what they did, again this is from the context of fitness landscapes, what they did was the following. Imagine that this is the E. coli that we started with. After that, we evolved this E. coli in low glucose, for several thousand generations. And at the end of this several thousand generations, I have this evolved E. coli. Both obviously have their DNA. And I sequence the DNA to identify what nucleotide changes have taken place between these two strains at the start of the experiment and at some time t. And what I note is that in this time that has passed, 5 mutations have taken place in different parts of the DNA of E. coli. So, the number is exactly 5, and this should strike you as important because now we have a mechanism or we have an experimental setup to test the results that we saw previously from Daniel Hartel's group, that what happens to a fitness landscape structure when we have 5 mutations within a gene. In this context, these were the 5 mutations that were the first ones to happen in the evolution experiment. They are not confined to one gene. In fact, they are confined to 5 different genes spread throughout the genome, but we know their identity and we know their location and we know the genes in which they do and what sort of changes do they drive. So, the first thing we want to check is the relative fitness of these. So, we will draw a graph like the one we were drawing for MIC, whereas this is the start culture, the start bacteria, and this is the 5 mutation bacteria. And on the y axis will again be fitness. And now in the previous study we saw that fitness was not directly measured as a growth rate, but it was measured as MIC, that was a proxy for growth. In this case, since they are being evolved in low glucose environment for such a long time, their ability to grow or their ability to grow fast in low glucose is a measure of measure of fitness. So, what is being measured in terms of fitness is growth rate in low glucose. So, obviously, as this population is being continuously involved in these low glucose conditions for a really long time, it is going to get better and better adapted to that environment and beneficial mutations will be acquired. So, at the start of the experiment, if this was the fitness, after being propagated for several thousand generations, fitness would increase to a higher level. And the question that we ask is the same question that Hartle and co-workers asked, that now we have the same scenario where we are moving from 0 mutation to 5 mutations, and hence by the logic that we used in the previous study, there are 120 ways to move from 0 to 5 mutations. How many are adaptive or what we called permissible, and how many are not allowed by natural selection? Remember, in the previous case, we saw that these numbers were permissible were only 18 and non-permissible were 102. If this study also sheds light to the extent that we get similar sort of numbers, then we are beginning to see generality in an evolutionary process. However, if this study tells us numbers which completely switches them on its head, then we are sort of lost as to, in some cases it is giving 102 non-permissible, and in some cases it is giving completely opposite numbers. And we'll begin with a discussion of what was seen in the results of these experiments in the next video onwards.